Hello everyone, welcome back to the NPTEL course, Experimental Biochemistry. This is the last lecture, and in today's lecture we will be talking about blotting techniques. We'll include Western blotting, Southern blotting and Northern blotting in today's class, and after that we'll be discussing MCQs and MSQs like always. So this is the last class and I will hope that you watch the video until the end. Okay, so let's begin. This is the NPTEL course Experimental Biochemistry, coordinated by Professor Dasgupta and Professor De from IIT Kharagpur. I am Beas Mukherjee, a research scholar from Dr. Shomo De's lab, and today I will be discussing with you the upcoming topic, which is blotting. Okay, so let us begin. So first let's start talking about blotting. So what is blotting? It is actually the process of transferring macromolecules. Now what are macromolecules? Of course, your protein, DNA and your RNA. So nucleic acids, proteins, these are all macromolecules, right? So they are transferred from a gel onto a solid surface or an immobilized membrane to detect the molecules that have been transferred. So blotting techniques have a wide range of applications in the molecular biology, genetics, biochemistry, biotechnology, medicine and different fields, okay, of science. So what happens in blotting? Let's find out. So you can see in this figure, the first step is protein extraction. So here they are talking about Western blot because blotting for proteins, it is known as Western blot. So we will learn all of these names very soon. So what happens? Let's see. So the protein has been extracted and SDS page has been carried out. So we have learned about SDS page in our previous lectures. Okay, you can go through that if you want to recall, and then after that Western blotting is performed and then we can see the result. So to confirm whether this is the specific protein that we are dealing with, Western blotting is done. It has a lot of functional assays. Okay, it's a very sensitive technique, a very useful, very powerful technique, and you can study a lot of different phenomena using blotting. Okay, so blotting techniques such as Western blotting are extensively utilized in molecular biology to detect and quantify specific proteins in complex mixtures, aiding in understanding of cellular processes and disease mechanisms. In forensic science, DNA blotting techniques like Southern blotting play a crucial role in identifying various variations, genetic variations, and establishing DNA profiles, supporting criminal investigators and paternity testing. So if you have heard about this RFLP and RAPD, you will know that blotting, actually Southern blotting plays a very important role in forensic investigation. Okay, to identify the specific DNA of, let's say, the criminal. Okay, or in paternity testing where you can identify the specific DNA. So for that, blotting of the DNA and then complementary binding with the probe of the complementary sequence hybridization that is carried out to confirm the specific DNA sequence present or not. Okay, so let's learn more about blotting. Right. So what are the different applications of blotting? So why do we even need to study about blotting? So here you can understand that blotting has applications in all fields, all disciplines of science. See, gene expression analysis. So determine the expression level of specific genes by detecting mRNA transcript. So for mRNA transcript detection, you need to go for northern blotting. Right. These are the names that are given. We'll come to it very soon. So for mRNA transcript detection, northern blotting. So whether a particular gene is expressing or not at a particular condition, let's say cancer cell versus your normal cell, what kinds of genes are getting upregulated, what are getting downregulated, that you can carry out, that you can find out using your northern blotting or quantitative real-time PCR. After that, we have protein detection and analysis. So detect and analyze proteins, their expression levels, post-translational modifications and interactions using Western blotting. For the same thing, for studying gene expression, you can do it at two different levels. One is mRNA transcript expression, the other one is gene expression through protein analysis, protein studies. So for that, for proteins, you need to go for Western blotting. Fine. Then we have DNA analysis. So specific DNA sequences are identified using Southern blotting. Okay, to find out whether there are any genetic mutations or not. So using Southern blotting, you can do that. Then there is RNA analysis. Once again, RNA stability, alternative splicing, mRNA processing, all of that can be studied using your Northern blotting. Then we have glycoprotein analysis. Mm. So what happens here is that uh, glycosylated protein. So these are all post-translationally modified protein, right? So glycosylated proteins. Hello, welcome back. This is the last class and we are talking about blotting today. So we just started the class and we are looking at the different applications of blotting. Okay, so glycoprotein analysis, here what we do is we detect and analyze the glycosylated proteins and study protein. Hello, good evening to you. Okay, so protein glycosylation patterns using Eastern blotting. So in Eastern blotting, here what we do is we study about carbohydrates. Okay, so we will learn about all of that soon. And then there is protein-protein interaction. So what happens in protein-protein interactions? Again, your far western blotting. And then DNA protein interaction, this is studied using south and west. So southwestern blotting, right? South is for DNA and west is for proteins. So basically blotting was first discovered by uh, a person whose name was Southern, and that is why Southern blotting, that term came into play. After that, with time, different kinds of biomolecules started getting blotted, okay, and then it actually became Northern, Western, and all of these different kinds of blotting, okay. So again, biomarker identification. So identification of a specific disease, prognosis, diagnosis, monitoring using blotting techniques. Then there is drug development. So here drug protein interactions and drug effects on gene expression, all of these you can study using again blotting techniques. For forensic analysis, this is very interesting. So for DNA profiling, for that southern blotting is performed. And then identification of genetic markers, which I was talking about, your RFLP and RAPD, right? And analyzing DNA evidence in criminal investigations. So for all of these purposes, blotting is very, very useful. Now, we will look at the different types of blotting. Okay, so see, northern blotting, that is for RNA. So here the different citations are given. If you want to learn more about these, the people who in which first time it was talked about, those papers are all cited here. Okay, then we have north and western. North is for RNA, western is for protein. So this is RNA binding protein, right? That blotting is carried out. We have far western blotting. Far western means protein-protein interaction. Western blotting, protein. Southwestern, protein DNA interaction. EMSA, once again, protein DNA interaction studies. Okay. And then we have southern blotting, it's definitely DNA. Eastern, western blotting. So that is, eastern is for lipids. Okay, eastern blotting is for lipids and yeah, so that is for lipids and other post-translational modifications of the uh, molecules, biomolecules. So lipid and protein, that is eastern western. And then we have eastern blotting. Eastern is for post-translational modifications. Middle eastern. Middle eastern is for DNA and RNA, and far eastern is for lipid and post-translational modifications. So these are the different names given to blotting techniques. You can read about these. These are very, very interesting and you can learn more about these techniques. So right now we will have a look at the most common blotting techniques. Okay, what are they? They are southern blotting, which is done, the target molecule is definitely DNA and specific DNA sequences are detected. You can study the copy number of DNA, you can study the genetic mutations using this, and you can study the fragment size, okay, which is useful in restriction fragment length polymorphism. Then again we have northern blotting, which is carried out for RNA, and this is used to detect specific RNA sequences and analyze gene expression, study mRNA processing, investigate RNA splicing activities. Okay, for western blotting you have detect specific proteins, analyze protein expression levels, identify post-translational modifications, study protein-protein interactions, and so on. For eastern blotting, you have glycoproteins are detected in this case. Okay, and then for far western blotting, you can understand protein-protein interactions are studied using labeled proteins as probes. So what are probes? We will study very, very soon what probes are. Then there is southwestern blotting, So for southwestern blotting, DNA binding proteins are detected. Okay. Now we'll move on to, okay, now this is very important. What is dot blotting? So dot blotting is a technique in which the transfer process can be omitted. So we will look at this, and this is a very useful technique. This is used for DNA, protein, antibody, small molecules, and so on. Okay, so 1 second. Now, qualitative analysis of biomolecules to find out whether a particular biomolecule is present or not, dot plotting is performed. Okay, so you can see this small diagram is going to make it easy for you. The proteins are detected using Western blotting. DNA, presence of DNA molecules, the mutations in DNA, fragment length, intensity, everything is detected using Southern blot, and RNA is detected using Northern blotting. Okay, so again this is a small table. This is telling you the differences between northern, southern and western blotting. So we can go through these at a glance quickly. So northern blotting, again you know, it is useful in detection of RNA, right? And southern blotting DNA and western blotting proteins. Fine. So specific sequence is detected using both, all the three. Correct. Now in all of these you can see that usually capillary transfer is carried out in case of southern and northern blotting, but in case of western blotting we go for electroblotting. Okay, so we will go through the techniques. You will learn about capillary transfer and electroblotting also very soon. Then cDNA or RNA probes are used with radioactive or non-radioactive labels in case of RNA blotting. For DNA blotting, you have single-stranded DNA or RNA as a probe, which is again labeled. Okay, but for proteins, you have antibodies against a specific antigen. Right, epitopes are used as probe. Fine. What are the other differences? Let's go through. Okay. So detection mechanism. For northern blotting, the detection mechanism is chemiluminescence and X-ray film. For southern blotting, same chemiluminescence and X-ray film. Why? Because radioactive isotopes were there, or fluorescently labeled isotopes were there at the probe. Right. So if the probe is binding, if it is hybridizing with the gene of interest, then it is going to glow and you will be able to see it. Otherwise, it will get washed away. Right. There are a lot of washing steps in between. So detection mechanism including CCD camera, film, LED or infrared imaging mechanism. So in this case what happens, there is a chemiluminescent substrate in case of Western blot. So this substrate is basically colored. Because of that they are using these CCD camera, LED, etc. So this was developed in 1979. That is your northern blotting. You can see southern. This was developed by Southern in 1975, and in 1979 also protein blotting was, Western blotting was developed. Then northern blotting encompasses denaturing formaldehyde agarose gel, southern blotting agarose gel electrophoresis, and protein blotting is done using SDS page. It can be used for gene expression studies and assist in disease diagnosis. I am sure you have already understood the applications of different kinds of blotting, right? So this you can read by yourself. So yeah, for southern blotting, you are having this paternity testing, victim identification, DNA fingerprinting, and protein blotting can be done for a lot of different kinds of protein studies, right? So now we will move on to the steps involved in blotting. So this is very interesting. We have already got into the core area of blotting, which is what is done in blotting, right? So first you can see there is this biological sample. Now this biological sample is having your DNA of interest, it can have your RNA of interest, it can have your protein of interest. Now here they have separated using gel electrophoresis. So here they are showing SDS page. So this makes us assume that this is definitely Western blot that we are talking about. Okay, so Western blot is also called immunoblotting. Why? Because antibodies are used in the detection and antigen epitopes are detected in this case, right? So here you can see that this is basically a setup in which a protein of interest is going to be detected. So firstly, exactly how agarose gel your polyacrylamide gel electrophoresis is carried out, that is carried out. Okay. So see, electrophoresed agarose or acrylamide gel, and then we have the dye front, and then we will run it through the sample. We will run the sample through agarose gel electrophoresis. After that, what we see is transfer from gel to membrane by electric current or capillary action. So if it is done using electric current, that is called electroblotting. Otherwise, it is called capillary blotting. So what happens here? Basically, after you have run the gel is having, you can see here that there are different proteins right inside the gel, but then you cannot see them unless you stain them. So here what we do is we do not stain it. We can check whether the proteins are present or not, and for that we can use the Ponshu stain. Okay, so what is a Ponshu stain? Ponshu stain, it can be very easily instantly stained using Ponshu, and it is a red color stain that tells you whether the proteins are there or not. Then what you do is, then you wash it and then you go for the transfer. So here what happens, basically what blotting is, your gel is placed, on top of the gel a membrane is placed. Now this membrane is often nitrocellulose membrane. So nucleic acids have a propensity of sticking to cellulose membrane, right? So what happens when you pass electric current? So there is a lot of filter paper below the gel. There is a lot of filter paper on the nitrocellulose membrane, and your buffer is running all through. Okay, it is very well connected by a buffer system. So what happens? You run electric current. It passes through the um, filter paper, it passes through the gel, it passes through the nitrocellulose membrane and onto the another filter paper, and then to the electrode. So from this electrode to another electrode, your electric current is passing. Now what happens? The proteins, basically what happens is, these proteins, they actually migrate from the gel to the nitrocellulose membrane based on the charge in the protein. So proteins are definitely negatively charged here, and therefore they are going to travel towards the positively charged anode, right? So that in that exactly same way, everything is the setup is done so that it migrates towards the nitrocellulose membrane. So now we will look at the entire process in details. Okay, so typical blotting experiments involves 4 steps. So give me a minute. Hello. আচ্ছা ঠিক আছে, আমি কাউকে একটা বলে দিচ্ছি যেতে। পেমেন্ট হয়ে গেছে তো? হুম, আচ্ছা ঠিক আছে, ঠিক আছে। আপনি একটা কাজ করুন, আমি নাম্বার পাঠিয়ে দিচ্ছি, তাকে কল করে নেবেন। ঠিক আছে? হুম, 8910 1000083। হ্যাঁ হ্যাঁ হ্যাঁ, ঠিক আছে ঠিক আছে। না, ওটিপি তাহলে দিয়ে দিন। হুম হুম, আচ্ছা ঠিক আছে, হ্যাঁ হ্যাঁ ঠিক আছে। Okay, so we'll carry on. Okay, so what happens in a typical blotting experiments? This involves 4 different steps. Okay, so gel electrophoresis of the sample that we looked at, and then we have transferring or blotting of the electrophoresed sample from the gel onto a membrane. Then treating the membrane with reagents or probes. So what is this probe? Probe is the single stranded DNA or RNA which is going to bind to your RNA of interest or DNA of interest, or it is the antibody which is going to bind to your protein of interest, right? Then what happens, and it is very specific for this target biomolecule. Okay, so it is going to bind, and that step is called probing. Then after that, developing the membrane for visual detection. So the probe is usually radio labeled. It can be fluorescently labeled. Okay, in Western blotting, there are two antibodies. One is a primary antibody which binds to the antigen using its paratope, and then there is a secondary antibody which binds to the primary antibody's Fc region. Okay, it is complementary to the Fc region of the second of the primary antibody, and so it binds, and then there is a whole new reaction going on. So we will study about that in details now. Okay, so this is basically the small gist of what happens in a blotting process. So see, one thing to note is that there is a blotting technique that does not involve electrophoresis. So this is called dot blot. So a dot blot is a technique in molecular biology and immunology to detect and analyze the presence or absence of specific biomolecules like DNA, RNA, proteins, antibodies, small molecule, etc. Okay, and so it is a qualitative method that does not involve gel electrophoresis, making it relatively quick and a simple method compared to other blotting techniques like southern or northern or western blotting. So in a dot blot, the sample containing the biomolecules of interest is spotted or dotted onto a solid support membrane, typically nitrocellulose membrane or PVDF membrane. PVDF is polyvinyl polyvinyl leadine difluoride. The sample is applied in small dots onto the membrane, and then it is fixed onto the membrane, and then fixing the spotted biomolecule onto its surface. Okay, so that is dot blotting. Then we will see what happens next. Okay, so you can see this is the gel, and here your sample is supposed to be there, and then you place the membrane. Then what happens? Membrane transfer takes place, and your entire sample now comes onto the membrane. So how do you know whether the transfer has happened or not? You can see the ladder here. This ladder is visible, right? You will be able to see the ladder in the membrane too. So nucleic acids and proteins and samples cannot be seen with the naked eye, but then the ladder can be seen because it is colored. Correct. So you will see the ladder. Then this transfer step is basically done in two different ways. One is called capillary blotting and another one is called electroblotting. So let's look at these in details. With capillary blotting, the experiment is arranged so that the membrane is placed on the top of the electrophorese gel. Dry paper towels are placed on top of the membrane along with a weight to aid in the blotting process. So along with the liquid buffer, technically called transfer buffer, the electrophoresed macromolecule samples move from the gel to the membrane by capillary action. However, once okay, so see, however, once the actual samples reach the membrane, they get stuck to it and they cannot go beyond the membrane because of the paper towels. This is because the membrane is chemically modified to have a special affinity for the macromolecule, so that once the samples reach the membrane, they get stuck onto it. In the end of the capillary blotting process, the paper towels are soaked with transfer buffer and the samples remain immobilized onto the membrane surface. The transfer of gel to the membrane is complete at this point. Okay. But what happens in case of electroblotting? So electroblotting means blotting or transferring the sample from gel to the membrane with the help of electric current. Here is the working principle. So nucleic acids are negatively charged molecules. Also in SDS phase, the charge of the protein is negative due to SDS treatment. So what happens when negatively charged molecules are exposed to electric current, they migrate towards the positive electrode. This phenomena is called electroblotting. So following the process, membrane is placed on top of a gel and the gel membrane is sandwiched between two sponges, and these sponge gel membrane sponge cassette right, is placed between two electrodes so that the membrane is at the positive electrode and the gel toward the negative electrode. So the whole setup is immersed with plenty of transfer buffer for the electric current to flow properly. Due to the electric current, the macromolecules migrate and are transferred from the gel to the membrane. So I'm sure you've understood this. Then comes in the process of probing. So you can see that probing, it's done by hybridization. So if this is your probe is basically labeled. Probe is exactly complementary to the sample of interest. And so probe is being pipetted onto the buffer solution for binding of its target. So this is how you're giving the probe. You're just applying using a pipette. Then what happens? The electrophoresis nucleic acid or protein samples which cannot be seen using naked eye, membrane embedded within buffer solution. Then we have the next task, which is hybridization. So in nucleic acid blots like northern or southern blot, there is a single stranded DNA or nucleic acid. Okay, so DNA or RNA that is added and then it goes and immobilizes. So there can be non-specific bindings as well, and for that we have the washing step. Okay, it is constantly washed with buffer. So single stranded nucleic acid probe binds to the nucleic acid, it gets immobilized, and then what happens? Just to put things in perspective, oh, this is given. This is, I'm sure you know that A and T binds and G and C binds, fine. Then for protein blots, Western blots and its derivatives, the probe is a primary antibody. So what I was telling you is that this is the probe. Probe is basically your primary antibody which is complementary to the protein. The protein is the antigen. Probe binds to the protein using its FAB region, and the secondary antibody binds to the probe FC region. Okay, it binds to the primary antibody's FC region. And this secondary antibody is often coupled with a enzyme, right? This enzyme acts on the substrate and it gives you the colored product. So when there is the presence of the antigen on the membrane, it is going to give you colored product. Okay, now there comes in some other steps. So for example, if you are using nitrocellulose membrane, you need to make sure that the sample is getting stuck to the membrane. It is fixing to the membrane. Okay. For that, denaturation is done and it is heated to a very high temperature to make sure that it is stuck to the membrane. There is also some buffer provided to make sure that it is sticking to the membrane. Okay. So after that, the probe is added. After hybridization is done, we need to make sure that there is no non-specific binding. So to make sure there is no non-specific binding, it needs to be washed. And it also needs a blocking step. So blocking is done with BSA, which is your bovine serum albumin, right, in Western blot. Now see what happens. Treating the membrane with probe that binds to the target molecule. Probe is being pipetted in the buffer solution, fine. Probe gets embedded, and then before this there are a lot of different steps. Okay, what happens? There has to be multiple washes, and then you are adding primary antibody, you are adding secondary antibody, multiple washes are done, and then it is incubated overnight. After that you will be able to see the image for Western blot and sample image for Northern blot. So this takes a lot of time, and if you start today afternoon, you are going to complete this by the next day afternoon or morning. Okay, so how do we see the signal? Just now we saw in the figure that we could see the signal, right, in Western and Northern blot. So how do we see the signal? Developing the detection signal. So developing the detection signal for actual visualization of the target of the sample is the final step in any blotting experiment. Simply put, the membrane is chemically treated so that the probe-bound target shows up on the membrane. This ensures that the experimenter can actually see it and draw inferences from that signal. For visualizing the target on the membrane, the probe has one of the following detection systems associated with it. So one is colorimetric, which is based on color. Okay, so the probe is tagged with an enzyme, which is often horseradish peroxidase, and this enzyme acts on its substrate. It breaks down the substrate to give a colorful product. Okay, so fluorometric detection. Here what happens, the probe is tagged with a fluorophore, and what happens when you visualize it by giving specific wavelength of light, it emits fluorescent light which you can then check. Okay, you can determine from the fluorescent light exactly how much sample is present in your solution. Then we have the chemiluminescent detection, in which the probe is stacked with an enzyme. The enzyme is treated with a substrate for this enzyme, and it is broken down by chemical reaction that emits light or luminescence. This emitted light is the qualitative and quantitative readout of this target. Finally, and the most sensitive method is radioactive detection. The probe is tagged with radioactive atom. Technically, this is called radio labeling. In nucleic acid blot, it is often a P32 radioactive isotope of phosphorus added in the sugar phosphate backbone of the nucleic acid probe. The probe, because of this radio label, gives out radioactive radiation, and that is captured using X-ray film. Okay. Finally, I'm sure you've understood the entire process. This is your probe. Probe is either radio labeled. See here, it is labeled with P32 isotope, and on X-ray film you can see the small black dot, which is the presence of this particular protein of interest or DNA of interest. In this case, it is RNA of interest, right? Because you can see U here, right? Specific RNA of interest is C. Now you can go through the entire process of southern blot. Let's have a look. So this particular genome is here. You have digested using restriction enzyme. So what happens? Small fragments. This particular tiny fragment contains your gene of interest. So DNA containing the gene of interest is extracted from human cells and cut into fragments by restriction enzyme. Then what happens? Then there is agarose gel electrophoresis. So the fragments are separated according to the size using agarose gel electrophoresis. Then there is, you can see the third step, that there is a transfer process. So here what process is used? This is a capillary blotting, right? So DNA bands are transferred onto a nitrocellulose membrane by blotting. And then what happens? They are positioned exactly on the NCM as they were positioned in the gel. Then what happens? It is exposed to radioactively labeled probe for specific gene. The probe will go and bind, it will hybridize with the sequence. And then what happens? If you wash it, remove the non-specific interactions, and then expose it to X-ray film, and you see the gene of interest intact there. Correct? So this is the second last slide for the theory portion. Here you can see in details Western blotting procedure. So SDS page is carried out, sandwich blotting that is performed. You have electroblotting here. You can see that the gel, from the gel to the membrane, which is PVDF in this case, all the your proteins are basically migrating, right? And then we are blocking the membrane with BSA or milk casing. This is very interesting. To make sure that there are no non-specific interactions between the probe and the proteins of interest, we are blocking it using BSA. Then what happens? The primary antibody protein interaction happens. Then you are adding the secondary antibody. Secondary antibody is containing the enzyme, and in this case it is horseradish peroxidase, which is an enzyme. And then what happens when you give the substrate? It is going to act on the substrate. It is going to give you a product which is a chemiluminescent product, and then through autoradiography you can see, right, the presence of, there is an X-ray film and you can see the different bands. So this is the entire story of your Western blot. So now we will try to see, I am sure that you are curious, exactly what chemiluminescence reaction is going on, because of which we are able to see the protein of interest. So this is the last slide for our theory part, and this is a very interesting slide because this tells you exactly what chemical reaction is going on between the enzyme and the substrate that is making us able to see the protein of interest in the gel. in the NCM or PVDF. So here what happens? Oxidation of luminol. Luminol is a substrate. Okay. So luminol gets oxidized using hydrogen peroxide in the presence of horseradish peroxidase, and a particular substrate, different products are getting formed. Okay. Amongst them, light is one. So H2O2 is the oxidizing agent here, and luminol is a derivative of phenyl hydrazine. You can look up the chemical formula and the chemical structure if you want. So HRP is used in Western blotting due to its high efficiency in catalyzing peroxidation reactions, and so the excited state of this 3-amino phthalate subsequently returns to the ground state, emitting light in the process. So excited state substrate is formed. The electronic transition results in emission of fluorescent light, okay, chemiluminescent light, so allowing for the detection and quantification of the target protein in the Western blot. So that is all. Now we will look at the different questions. So this should be assignment 12, sorry. And we have all questions of this assignment. So let us begin with the questions. You can answer in the chat box like always. Okay, I'm keeping the chat box open. Okay, so the first question is, what is the first step of Western blot? And the second question is, Western blotting is performed to detect what? Specific DNA, specific RNA, specific protein. You can tell me. The questions are very, very simple, and we are going to complete this very, very soon. By 7:30 we should be done with this topic. Okay. B, very good. Running an SD speech, very good. And the second one is C. Yes, very good. That is absolutely correct. So we'll move on to the next two questions. Western blotting technique is known, also known as, and the proteins in a sample are separated using what? C, immunoprecipitation. And then you're saying it's B. Okay, let's see. Let's look at the answers. No. It's actually Western blotting technique is also known as immunoblotting. Why is it called immunoblotting? Because we are using antibodies, right, in the process. For blotting, we are actually using antibodies as proof, and that is why it is called immunoblotting. And how are the proteins separated? They are of course separated using SDA speech. Right. So we'll move on to the next two questions. Sorry. Okay, try to answer this now. For DNA fingerprinting, which of the following technique is used? You have southern blotting, northern blotting. Yes, very good. For the next question, in blotting techniques, a probe is a specific protein to detect DNA, a short piece of labeled double-stranded DNA which is complementary to a specific DNA, a short piece of labeled single-stranded DNA or RNA which is complementary to specific DNA or RNA. C, yes, that is very correct. That is correct. Okay, now we move on to the next question. So what technique is suitable for detecting specific mRNA expression in cell? Specific mRNA expression. B is not in blotting. Okay. For the next question, we have the non-specific sites on PVDF can be blocked using B. No, you haven't answered yet. Okay. B. Okay. Let's look at the answers. Okay. Very, very good. Both of these are correct. And then we have the next two questions. Why the probe is labeled, and why is the probe labeled? To improve binding capability, to improve location identification, to improve visibility to improve stability. So you're saying it's location identification. Okay. So what is the major force behind protein or nucleic acids to go out from gel onto the membrane? Because of what are these protein molecules or nucleic acids traveling from gel to the membrane? A is electric current. Yes, yes. Let's look at the answers. Very good. So yes, it is electric current, and if it is an MCQ, it can also be voltage, but I think an appropriate answer should be electric current, right, and voltage both. Okay. So why labeled antibodies are used? To detect the presence of particular DNA, to detect the particular RNA in southern blotting, protein in western blotting, DNA in, okay, protein in western blotting, good. And then which of the following is or are used to detect protein in western blotting? B, antibody, very good. Okay, very good. Which of the following membranes is best if you want to strip an already used membrane for western blotting? So now they are asking you about membranes. Okay. So stripping. So B is the right answer. PVDF. Yes. So if you want to reuse membranes, PVDF is a better option. We will soon find out why. Why ghost bands appear in blots? Too much of protein has been loaded, blocking was not done, incubation with primary antibody was too long, too much substrate for secondary antibody was added. So that means too much of primary antibody was added. So which one do you think is the case? A, B and C, right? So ghost bands can be because of a lot of different reasons. So too much, okay. Okay, so when too much protein has been loaded, ghost bands can appear. And you're saying that it is A, B and C. Blocking was not done. Incubation with primary antibody is too long. You can look this up. Okay, you can try to check if this is correct or what you are answering is correct. Okay, you can search what ghost bands are and why they can be formed. Okay. And for this one, for the previous answer, it is absolutely correct. PVDF is better for stripping and reusing. Now we will look at the difference between PVDF and NCM. Okay, so PVDF is useful for it has very high protein binding capacity and useful for proteins of varied molecular weight, including the ones with low molecular weight. So they have high mechanical strength and chemical resistance, and therefore stripping and reusing is possible using these PVDF membranes. Now, PVDF membranes are compatible with various detection methods, including chemiluminescence and fluorescence. NCM, however, have high protein binding affinity and generally are more cheaper. Okay, they are suitable for proteins of moderate to high molecular weight and are commonly used in routine protein blotting applications. NCM are more prone to degradation under harsh conditions and therefore cannot be stripped. So, PVDF membranes offer advantages in terms of versatility, durability, and compatibility in various detection methods. Okay, so now there is the last question, which is if you forget washing the membrane after incubation with primary and secondary antibody, what will happen? If you forget to wash, then what is going to happen? A, background noise will be very high. Okay, let's see what is going to happen. Okay, A, B and C, everything is going to happen. Your background noise is going to be very high. Whole blot will be dark. Okay, and no specific band will be observed. So, these are the different places from which I have collected all the information. All the best for your exam. Keep calm and do your best. Thank you for attending all of these lectures all these days. Thank you so much. Oh, very good. You've answered also B and C. Thank you. Thank you so much. If you have any questions, you can ask me. Otherwise, I'll end the lecture now. Thank you so much for attending and good luck in future. Okay, if you don't have any more questions, we can end the session now. Thank you. You too.